Okay, good afternoon, everybody. So I'd like to start session 13, the resistance to the antifungals and the other antimicrobials. And this session we have the two talks. The one is the invited talk and one oral presentation. My name is Ryo Honda from Kansas University. I chair with this session with Jimmy He. So I would like to invite the first speaker, Johanna Rhodes. She completed her PhD in the host gene regulatory networks activated response to the fungal infection at the University of Warwick, and Jo moved to Imperial College London to study pathogens and focus on human infection. And her research has focused on 3 or 4 WHO critical priority group fungal pathogens, including Cryptococcus, Candida, Aspergillus. And she's now at the Radboudumc in the Netherlands as a PI, and her research group uses One Health approach to balance and optimize the health of humans, animals, and the environment. So please welcome Jo. Jo, floor is yours.
Hello, everyone. So thank you for inviting me to speak at this conference. I realise that this session is perhaps a little bit out of the ordinary from what you've been listening to over the last sort of nearly 2 days. Hopefully, you're going to see that there's a lot of similarities between antifungal resistance and antibiotic resistance. I've been quite inspired by the talks that I've seen, the posters that I've seen. I sort of look at stuff and I go, oh, that's really interesting. We see that in fungi. And also, I've had so many ideas as well by looking at some of your posters and thinking, does that possibly happen in fungi as well? So it's been really encouraging to see, and I've got lots of ideas, so hopefully this will give you some ideas as well, potentially of how we can work together to look at antimicrobial resistance, antibiotic, antifungal, and otherwise. So thank you for that lovely introduction. We're going to be talking about evolution of antifungal drug resistance in pathogenic fungi. I'm going to focus on a particular fungal pathogen called Aspergillus fumigatus. So fungi are ubiquitous. Today, you have already breathed in several fungal spores. Some of them are probably drug resistant as well. But I'm pleased to say that if you have a fully functioning immune system, you're okay. If you do not, if you're immunosuppressed or if you're immunocompromised, then that's not so great for you. And fungi are opportunistic pathogens, and they can cause quite serious infections. So fungi and animals diverged from other eukaryotes around 1 billion years ago, and then the fungal and plant kingdoms diverged about 600 million years ago. There's thought to be at least 150,000 fungal species, but recently this is estimated to be a lot higher. So this paper that came out in December last year that says 2.5 million, this is a really good estimate. And the reason that this number has jumped so much is because of all the metagenomic data that's being generated and this so-called dark taxa, when we've realized that we've massively undersampled and we've underestimated what we know about fungi, and there are a lot more fungal species than we thought. So over 8,000 cause disease in plants, so the staple crops, wheat, barley, etc. But then also the things that we really enjoy, the things that we kind of need to get through our day, like coffee, chocolate, bananas, all these things that we really enjoy are under threat from fungal disease, decreasing yield, and impacting our food security, or shall I say food insecurity. And over 600 are associated with humans as commensals or pathogens. So when we think of fungi, I probably think that a lot of us kind of think of this fairy tale red toadstool with the white polka dots, Snow White skipping through the forest, and that is exactly correct. Like, that is a real fungus. But it's only a part of a fungus. It's what's called a fruiting body, and it's a certain part of a fungus life cycle. And it's a certain part where they are undergoing perhaps a sexual life cycle. They can also undergo an asexual life cycle, a parasexual life cycle, bisexual, unisexual, you name it, they do it. And fungi are all over the place. They generate spores, so they're in the air. They generate hyphae and mycelia in the networks. They can be water molds as well. They're on that punnet of strawberries that you've forgotten about that's in your fridge at the moment. They're everywhere. So fungi kind of break up into 4 main classes. You've got the Chytridomycota and the Zygomycota, which are kind of the first sort of basal fungi. And then recently, or more recently, shall I say, you've got the Basidiomycota and the Ascomycota. And these are the ones where we kind of get the pathogenic varieties. Over recent years, we've seen an increasing number of fungal outbreaks. So we've seen mass amphibian population declines in Europe due to Chytridomycota. We've seen decline in the bat populations in North America due to this white nose syndrome. So what happens is this fungus just grows over the bat's face. It doesn't kill it, it just interferes with the echolocation, so they can't find food and they just starve to death, which isn't very nice. It's thought that it was introduced by spelunkers from Europe. So all these lovely new activities that we're doing, perhaps we should just stay indoors. And then, of course, we've got the plant diseases as well, which are targeting, ash dieback, things like that. And then we've got the human disease. So we're seeing not just an increase in plant diseases, including crops and human, but also animals. I see reports on a monthly basis of penguins in a zoo in Belgium that are infected with a drug-resistant fungus, etc. So it's zoo animals, it's pets, it's farm animals as well. So we're really interested in finding out why. Why has this increased over the recent decades? And the short answer is it's us as humans. There's an increasing public health threat as well. So this is due to new emerging at-risk patient cohorts. So COVID has a lot to answer for. You may recall in the end of 2020, I think it was, there was this second wave of COVID that was going through India. And then about 6 weeks later, you had this wave of this black mold called mucor just going through all the people who'd had COVID. Well, that's because they'd had COVID, they'd been some sort of susceptibility to fungal infections. But then you can also have COVID-associated Candida auris, COVID-associated Aspergillus fumigatus as well. Certain patient cohorts undergo certain drug treatments, such as ibrutinib, they are at risk. You've also got new patterns of drug resistance caused by the dual use of antifungals. What do I mean by this? We're using the same drug classes to treat patients that we also use in agriculture and animal husbandry. Does that sound familiar? So it's not just an antibiotic issue, it's also an antifungal issue as well. We also see the emergence of highly fit resistant clones. These are the types of fungi where I go, right, okay, they're not just resistant, they are Steve Rogers after they took the super soldier serum injection, right? They are super. And you've got opportunistic infections becoming increasingly resistant to the first-line drug of choice, which is the azole drug class. You've also got 3 other drug classes where we're seeing emergence rapidly occurring. So the echinocandins, the pollines, the pyrimidine analogues. We've had a similar bottleneck in the development of antifungal drugs to, as we see in antibiotics. There are some new drug classes that are in clinical trials, phase 3 at the moment. But what we see is that fungi that are resistant to azoles are predisposed to develop resistance to other drug classes, including the new ones that are currently in clinical trials. This is incredibly worrying. So, with all this depressing news in line, the WHO in 2020 released its first ever fungal priority pathogen list. It divided it into critical, high, and medium priority pathogens. At the top, you've got 4 in the critical priority. You've got Cryptococcus neoformans, which is a yeast that causes a type of fungal meningitis in HIV/AIDS patients. Mortality is up to 61%. Candida auris, which is kind of the new kid on the block, it emerged sort of about 20 years ago and has just spread over every continent apart from Antarctica. Invasive infection, mortality up to 53%. Drug-resistant Aspergillus fumigatus, the mortality is up to 88%. Candida albicans, similar to auris, the invasive mortality is up to 50%. And there's a large number of drug resistance mechanisms in fungi. It's very different to bacteria in some respects, but actually I'm hoping this slide will make you realize that there are some traits that are very similar. So first we have aneuploidy. So here, 15 years ago now, Edward Sionov showed that in Cryptococcus neoformans, which is the same group of fungi as the red toadstool, they cause the fungal meningitis. He saw that if he grew Cryptococcus neoformans on agar containing fluconazole, which at the time was the drug of choice for treating fungal meningitis, there was a duplication in the chromosome that contained the gene ERG11. Now, ERG11 produces a protein that is the target of azoles. When he took those resistant colonies and plated them onto agar that didn't contain fluconazole, the aneuploidy was lost. So it was a selective pressure. Fluconazole was selecting and inducing the aneuploidy. We also see heteroresistance. Up until 10 years ago, no one really thought heteroresistance in fungi was a thing. Obviously, it's a thing in bacteria. I don't need to explain to you that heteroresistance is where there's a resistant minority population in a larger population. They are resistant, have the drug treatment, and then this minority population are resistant, and they expand and they grow. We see this in Cryptococcus neoformans as well when we have monotherapy, and our results showed that we can't treat with monotherapy anymore because of heteroresistance, and you have to use combination therapy with a different drug class. My personal favorite is hypermutation. So this is a really interesting one because hypermutation occurs in pretty much every fungal organism. And I keep writing grants, and the feedback I get is, this isn't novel because it happens in bacteria. And I'm like, yeah, but fungi. So it's really frustrating. So if anyone wants to work with me on this, I'll be really grateful because I love it. So hypermutation, you have mutations in genes in the mismatch repair pathway. What we see in fungi is some of these mutations, they truncate the protein. So they're nonsense mutations. Some of them you have multiple mutations in multiple genes, just causing an amino acid change. And there's this epistatic effect, and it just sweeps together and causes an accelerated mutation rate. And basically what happens is you have the evolution of beneficial mutations that might include resistance. And then finally, we have these amino acid changes in genes of interest. So ERG11 in Cryptococcus is called CYP51A in Aspergillus fumigatus. Rather annoying, but it's the same gene. And this is another mechanism that we see. We just see one nucleotide change causes an amino acid change, and it causes the binding pocket in the enzyme to get a little bit smaller. The drug can't fit in, therefore they're resistant. So this is what we see in Aspergillus. This is Aspergillus here, this kind of like this fluffy structure. It's a mold. And what we see is we see these amino acid changes. So it's a leucine to a histidine shift at codon 98, coupled with a tannin repeat of 34 base pairs. This tannin repeat causes an increase in expression of the CYP51A gene. The L98H causes the binding pocket to kind of seal shut. This causes resistance to itraconazole, a particular azole drug. And then this other one, this TR46, causes resistance to voriconazole, so two azole drugs. So we're really interested in how did these evolve, when did they evolve, where did they evolve, why, all the W's basically. So we're constantly exposed to airborne fungi. So here is Jen, she's the next speaker, setting up this Burkhardt trap, the roof of St Mary's Hospital in London, in an urban location, then also in a rural location. These are really cool because they filter the air 10 litres per minute. It just kind of deposits it on a tape and you can chop it up, do your PCR, do your sequencing, whatever. Found an abundance of Aspergillus fumigatus, And Aspergillus is primed for climate change. It can grow up to 60 degrees centigrade. No idea what that is in Fahrenheit, it's high. It loves compost as well because of all that mixing, all that heat that's generated, and it sporulates a lot. And the patient cohorts at risk of aspergillosis are as such. Cystic fibrosis is the top one. America has the largest cystic fibrosis cohort in the world, followed by the UK. And then you've got neutropenic transplant, HIV/AIDS to a certain extent, others, cancer, and then also recently we're seeing this severe influenza and also COVID-associated aspergillosis as well. So we wanted to see how much azole resistance there was in the soil, how much was in the environment. So here, we saw that actually azole resistance is in 7% of UK soil samples, which is a good baseline. But when you break it down into what sort of soils they were, we found something quite interesting. We expected the most resistance to be found in agricultural crops, and it wasn't. It was only 1%. We saw the majority of the azole resistance in urban flower beds, you know, after you've gone to the garden centre at the weekend and you've bought your tulips and your compost. We're like, oh, this is kind of interesting. So it's horticulturally driven, maybe. TR34L98H and also the TR46 mechanism as well. So that's kind of interesting. I'm not going to talk too much about this because Jen's going to talk about it next, but she looked at it in the air and she found it in the air. So fungi are ubiquitous, remember? This is in Vietnam, so this is a sample that we did in Vietnam, and what we could see was azole resistance was about 80% or higher in national parks, rice farms, fruit farms, shrimp farms, urban residents. It is everywhere, and also at concerning levels. So at this point in time, we thought, right, okay, we need to have a look at the genetic mechanisms underpinning this resistance. Because sure, we know that there's mutations on this particular gene, but what about the rest of the genome? This is an organism that has a genome size of over 30 million bases. It's huge. It has 8 chromosomes. It has a heck of a lot of recombination. There's a paper that came out last year that we did that shows it has the most amount of sex of any organism known. It has a lot of recombination. So Zain Chowdhury came to us and he did a little experiment to look at signatures of resistance to agricultural fungicides in our genomes. So we've generated the 1,000 Genome Aspergillus project. We've actually done 1,200 genomes, but who's counting? We've done a broad and deep sweep. So by broad, I mean we've sampled as many countries as possible. We've got a pretty decent worldwide distribution. We've also sampled temporally as well, so we've got isolates over the last 102 years. So pre-introduction of agricultural azoles as fungicides. And then deeply, so we want to sequence to a sufficient depth to call these variants accurately, even with recombination, etc. And this is the phylogeny here. It's a bit messy, it's a bit complex. There's no nice outbreak clonal lineage here. But what we can see is that resistance to agricultural fungicides, these ones in pink, they do tend to cluster on a particular genetic background, this sort of clade at the bottom. So this is resistance to a particular fungicide called benzimidazoles. And this is the beginning of a serious resistance problem to fungicides agriculturally. They've since been discontinued, but resistance can still be picked up in isolates that I sequenced last week. And what we can see is from these isolates, signatures of resistance to benzimidazoles is found in environmental isolates, but it's also found in clinical isolates taken from the patient. The only way the patient is going to have those signatures is if they've acquired that infection from the environment. We can also see the signatures of resistance to different agricultural fungicides as well, distributed worldwide. So what about the azoles? So these are the ones, these are the big ones. They're on the same background. So the TR34 mechanism is the blue. It's on the same genetic background-ish. You can see some blues poking out in the top one, and we think that that's because of recombination, likes to have sex, etc. But it seems restricted to this background. We don't know why. Well, I say we didn't know why. Why is resistance occurring? So we're seeing a change in agrochemical use, changing at-risk groups, changing climate, changing biota, changing virulence. But it's this dual use of fungicides, particularly the azoles, in agriculture, in horticulture, and in human treatment as well. And then we've got these industrial composters, which is just mixing in vegetable waste and vegetative growth from all these different locations. Aspergillus is meeting Aspergillus from 20 miles away and going, hi, it's like Bumble. And we are facilitating it. So the complex population structure of Aspergillus fumigatus actually starts to look a little bit simpler when you delve into it. Clinical and environmental isolates are derived from the same population as are itraconazole-resistant and itraconazole-susceptible isolates. And then when you look at how you can sort of divvy up these 1,200 genomes, it kind of roughly falls into about 5 clusters using sort of admixture type techniques. But what you can see is they're not clean. There's a lot of admixture going on, indicative of the recombination that's going on. If you were to do this on different fungal pathogens or even bacteria, you'd get quite nice, clean clusters. It's not the case here. And when you look at the presence of these 5 clusters globally, they are everywhere. You're not going to find one particular cluster in one particular geographical location. It is dispersed. And when you look at recombination levels, there are certain clusters that are undergoing more recombination than others. So when we map these clusters onto that lovely phylogeny that I showed a few slides ago, you can see that actually there is that signature of recombination. You can see the yellow isolates, they're kind of bleeding into the green is bleeding into the purple, etc. But then there's that blue one right down the bottom. It's only a few, but that is probably as close as we're going to get to a clonal outbreak lineage in Aspergillus. And what you can see is the first column is CYP51A, the target of azoles, and all of them have TR34L98H. They're resistant to itraconazole, but they're grey for the next 3, which are agricultural fungicides, which means they are not resistant to agricultural fungicides. The last column is MSH6, a gene in the mismatch repair pathway. It has a mutation in it. It has quite a few mutations in the mismatch repair pathway. So what's happened is the bottom clade, with the majority of the azole resistance mutations, have mutations in the mismatch repair pathway. They have an accelerated mutation rate, and some of them have moved onto this background, and they've just gone, and they're a highly fit resistant clone. The ones that are azole resistant and they've moved into the top clusters, they haven't spread because they haven't got an accelerated mutation rate. They've stayed constrained. So this is just basically doing the build-up. So these isolates, when you do Luria-Delbruck fluctuation assays, which are apparently a big thing in bacteria, didn't realise that until about 2 years ago, but they work in fungi, we can see this accelerated mutation rate in these isolates, up to 6-fold higher. So when and where did resistance evolve? So using a piece of software called Beast, and a lot of maths, and a lot of me banging my head against my desk for about the past 12 months, I have to say, we can see that TR34L98H evolved around 1980. Now, azoles have been around a lot longer than that, but in the 1970s, the chemistry altered to triazoles. It meant it was a bit of a tighter fit in the enzymatic pocket. So something about the introduction of that chemistry has made it almost easier for resistance to occur, for the selection to occur. So that's when TR34 has evolved. But what about where? So let me explain this. The darker the colour, the more likely it is that is the origin. So basically that nice dark blue dot right in the middle, well, it's in the middle of the English Channel, but it spreads over to East Anglia and then also to the Netherlands. What are the Netherlands famous for? Tulips! I swear I've got to go into witness protection after this because they'll kick me out. So the Netherlands are the biggest exporter of bulbs in the world, $1.5 billion. All the other countries, you can't even make them out because the Netherlands just dwarfs them. And they also use the majority, they use the most azoles in the whole of Europe at least. And basically what they do is they get these big vats and they get these tulip bulbs and they literally just dunk them in, they package them up and they ship them around the world. So they're spreading drug resistance. Buy a tulip. So think about that before you buy your tulips. The cat's already out the bag, it's fine. Anyway, so Aspergillus fumigatus is a WHO critical priority fungal pathogen, and it's got an 88% mortality rate up to. Azole resistance is primarily caused by this TR34L98H mutation. And it's hypothesized that the dual usage of azole antifungals in agriculture, horticulture, and in the clinic drive the resistance. There's lots of recombination in the population, but this mutation seems restricted somewhat. And this is coinciding with resistance to other agricultural fungicides and also the MSH6 mutations and the mismatch repair mutation. The timing of the emergence of this mutation coincides with the introduction of triazole antifungals agriculturally, and then the spatial evolution is in regions with high intensity farming. So I've really put the hammer on the Netherlands here, but I have to say East Anglia, if you don't know, there's a lot of fruit farms there, and they use azoles. It's just that the Netherlands uses more, so kind of leaning towards that. So really, we need to think about what we're using, especially when these new antifungals get the go-ahead. We need to basically keep some for clinical use and some for agriculture, and don't do dual usage anymore, else we're just going to end up in the same situation. Finally, this work cannot be done by it couldn't have been done without all these people working together on this, and my funders, the Wellcome Trust. And thank you for your attention.
Hi, I'm Connor Brown from Virginia Tech. Really interesting, exciting to see, especially here at this conference. Two things, one's a comment, one's a question. The comment is that I'm just reminded all this talk of recombination really sounds a lot alike the conversation we have about horizontal gene transfer in bacteria, sort of the co-occurrence of strains in an environment. And yeah, I guess that's just really interesting. And then number two is, do you think there is a role for eukaryotic mobile genetic elements in facilitating acquisition of resistance? Have you seen that?
Oh man, you've opened up a can of worms there. That is, it's a big topic. Yeah, so they have huge mobile genetic elements. We're talking like 20 kb sort of size, and some of them carry resistance genes, some of them decide whether they have sex or whatever. So that is a huge thing. And whole horizontal gene transfer, it's a relatively, we know it happens, but it's so understudied. And yeah, so it's on my to-do list as to how it's contributing here. Transposons, the works, yeah, but it happens.
Okay, thank you.
Excuse me, Lance Price from George Washington University. So in the US, we're seeing this rapid increase, heavy marketing of triazoles in agriculture, even commodity crops like corn and soy. And I guess this isn't a question, but a statement. One of the big differences, you use the analogy of food animal production, right? So they're promoting growth and preventing disease. Here's the same thing, right? But in the US, we had the FDA controlling or regulating antibiotic use in food animal production. They raised the bar about 20 years ago saying you've got to consider resistance in people. But the FDA, or sorry, the EPA is who regulates antifungals, right? So are there analogies in Europe? Are we seeing the same thing? Because they don't consider resistance. They only consider toxicity.
Yeah, completely.
Right? So we need to change this.
I completely agree. And one of the issues that we're having at the moment is that they're only really waking up to this, and there's not really sort of like a body that deals with this. And one of the things that we've come up against in the Dutch government is, oh, well, why should I care if it kills 100, if it kills 1,000 people a year in my country when there's something more pressing? And it's trying to get across the importance in the right way, because the farmers are willing to do something, just like they were with the antibiotic resistance. So it's just a matter of the right language, I think. Yeah.
You must have different farmers than we do.
In the Netherlands, they like to help out.
My name is Gustavo Ibaceta. I'm working for Health Science Now Research Institute. My question is a little bit technical. By the way, thank you very much. It was a great presentation. When you show the Bayesian tree, how you calibrate that? What did you use for calibration?
The phylogenetic tree?
Yes.
So we include, also say I include a very closely related species that we know is not fumigatus. So we basically try and force it to have sex, and it doesn't produce any viable offspring. So we kind of root it as an outgroup on that. So that's how we kind of calibrate it. But we've also got to take recombination into account. So what I didn't mention on the temporal analysis is you've got to strip out all the recombination, else it's just going to mess with your temporal signal. So a really good way to do that is to just use the mitochondrial genome, because there's less recombination going on there, or next to none.
Okay. Yeah. Thank you.
All right. It is now my pleasure to introduce you to the next speaker of this session. we are very lucky to be joined by Jennifer Shelton, and she is from the UK Centre for Ecology and Hydrology. And she will give us talk about citizen scientist surveillance of azole-resistant Aspergillus fumigatus in UK air and soils. So let's all give a warm welcome to Jennifer.
Thank you very much. Joe, smashed it. Great talk. Just really quickly before I actually start my talk, 10 years ago I joined the fungal epidemiology group at Imperial College London, and Joe was the senior postdoc bioinformatician and fellow there. She sat on the best desk space by the window, and I was incredibly intimidated by her. Sorry, Joe. But you've just seen her talk, right? You understand why. So it's quite something 10 years later to be in this session halfway around the world from Imperial, sharing this session with Joe and flying the flag for fungi. So that's cool. Thank you. Right, so I'm going to talk to you about my PhD, which is indeed using citizen scientists, not using, that's a terrible word, working with citizen scientists to surveil azole-resistant Aspergillus fumigatus in UK air and soils. So Joe, thank you very much for the intro to this. Okay. Appreciate this is a bit of an overwhelming slide, but Joe has already sort of covered this. So the bottom left, you can see a beautiful colony of Aspergillus. I think it's beautiful anyway. And I'm sure you've come across it plenty of times from that yogurt that you've left for too long in the fridge, or even when you peel your onion under the bottom layer, or if you have a food bin in your kitchen, don't have a food bin in your kitchen. There we go. When it lives in the soil, and essentially every spore head, which you can see in the top left, this is a diagram of what Aspergillus looks like when you look at it under the microscope. And also beautiful, you can tell I'm a mycologist, can't you? Each spore head produces thousands of spores every time it sporulates, and they just puff straight into the air, and we breathe them in all the time. So in this room, I probably just inhaled a couple of spores. If I was in the garden doing some gardening, I'd inhale quite a few more spores. So wear a mask when you're gardening. Anyway, that's the end of the story. So as Joe mentioned, if you breathe in these spores, if you're healthy, if your immune system is working perfectly, then it absolutely clears those spores and you know nothing about them. But unfortunately, if you're immunosuppressed, so if you're taking immunosuppressants or undergoing chemotherapy, or even if you've had a severe lung infection, such as flu or COVID, or you've suffered from TB or COPD, that innate immune system isn't going to work so well. Those spores can actually make it down into your lung cavities and sporulate and cause a range of diseases known as aspergillosis. And as Joe mentioned, that can have a case fatality rate of up to 88%. So just to introduce why azole-resistant Aspergillus is a problem, Aspergillus loves growing at human body temperature. So that isolate there was grown for 3 days on agar at 37 degrees, and it loves it. And unfortunately, when people do get aspergillosis, we treat them with azoles as the frontline drugs. We also spray azoles on our crops to prevent fungal diseases from plant pathogens. So there's this crossover, and what we're seeing is people getting azole-resistant aspergillosis infections who have never been treated with azoles before. So it has to be coming from the environment. So my PhD project was to look at the environmental drivers of azole resistance in fumigatus and its relevance to human health. And I wanted to find out what is the actual background rate of azole resistance in fumigatus spores in the UK? And is this different between air and soil? So the original plan, such a nerdy picture, was just me travelling around the UK in my beaten up Ford Mondeo, collecting air samples with a Burkhardt air sampler and digging little holes to take soil samplers. I submitted this plan after I'd already started to my 6-month assessors for my PhD and got that wonderful feedback that everyone loves from their 6-month assessment. This is not novel enough, what you're proposing to do. Wonderful. So, as you do when things are starting to unravel a little bit, I went on holiday.
And as I was sat by the poolside in Gran Canaria, thinking, how do I save my PhD? I was thinking about a postdoc at Imperial who'd come before my time that had used, you know, the sticky plate seals that you use in the lab to cover your DNA plates? He'd used them and he'd positioned them around St. Mary's, Imperial College, opposite the lab where Alexander Fleming discovered penicillium. And he captured on these sticky plate seals penicillium spores, and he used them to determine what species of penicillium Alexander Fleming isolated penicillin from. So I was thinking about these sticky seals, and I was also thinking about some really cool citizen science projects that people have done in the UK. So if you've not heard of these, Swab and Send and Beach Bums, just get on your phones now and have a look because they are super cool. Swab and Send just ask people to collect a swab from anywhere that they can think to swab, like the most disgusting places like their toilets or whatever, and send them in for antibiotic discovery. And Beach Bums is asking surfers to swab their bums and send them back to some unfortunate person who looks for antimicrobial resistance. And all of this was going around in my head as I was thinking, lying next to the pool in Gran Canaria. And this happened. Top left poster, that was the original Science Solstice, decided to run my own citizen science project because that might be a bit novel. You'll notice Stonehenge. Hopefully you're familiar with the connection between solstice and equinox in the UK and going to Stonehenge and smoking weed and hanging out on the field. So I figured that's a really good memorable date to choose for a citizen science project. So I put the poster together, put a few posters out around CEH and Imperial, sent it round, put it on Twitter. And my supervisors, Andrew in the front row there, and Matt Fisher, were like, Jen, honestly, people are just collecting mold, don't get excited. You're going to be lucky if you can get about 100 people take part in this. And honestly, I had to actually close it early because so many people signed up to take part. It's like, I don't think I can run this many samples. So yeah, I had 500 people sign up to take part. And then when it happened once, it was great. So I decided to repeat it 3 more times to get samples throughout the year. And then repeat it for soil, because with Aspergillus spores, what goes up comes down, or other way around. So you get the plate seal there that we asked them to collect the spores on, and also to collect little bags of soil and send them back to me in the lab. So this is just the map for how many samples were sent back for that first Science Solstice round in June 2018 for the air samples. So because I didn't want to put red, because red indicates bad, green was samples that did grow mold, and blue the samples that didn't. So actually that's quite cool in itself, because that's a sticky plate seal left out on someone's windowsill for 8 whole hours, and some of them didn't actually collect any fumigatus spores. But what I love about this is, you think of me driving around in my Ford Mondeo, I was literally going to do a little circle around Oxford, which is where I live, and this is the coverage that the citizen scientists got in a single day. Phenomenal. And because I'd launched it on Twitter and been very active with emailing people, people were absolutely great at sending me photos. They got their kids involved, which is really cute to see. If Uli's here, I'm pretty sure that's his sampler on the right-hand side. So yeah, people got really into it and were incredibly enthusiastic, given that it's mold. And this is what it looks like. Summer 2018, we got back 712 samples. I'm not going to go through and read all of these. You can read them. Autumn, 398. Winter, 321. And I just love on winter, someone actually went out on Christmas Day and collected a sample and sent it back and wrote a thing, family getting too much, grateful for the break. That's commitment to the cause. And then there was a bit of a resurgence come spring, and 465 samples. So in total, we got back nearly 2,000 air samples on 4 dates throughout the year, which together grew 2,366 fumigatus isolates. So the next step, once I'd picked each of those isolates into preservation solution, was to test them against tebuconazole, which is the third most agricultural azole, most sprayed agricultural azole in the UK. Just preempting a question, why did I only use the third most? Because tebuconazole's the cheapest of those top three, and I only had £5,000 to complete my PhD. So I pipetted each isolate onto a tebuconazole plate and recorded whether it grew or not to test its susceptibility. So for the air samples, you can see each round had slightly different percentages of azole-resistant fumigatus. So I'm just to explain, that's what ARAF stands for at the top, by the way, azole-resistant Aspergillus fumigatus. 3.8% in summer, 6.1% autumn, 3.1% winter, 9.1% spring. So I think there's something in that. I think it's quite interesting that it was variable between season. So overall, there was 111 isolates that were tebuconazole resistant, which is 4.7%. So if you think, on average, we're probably breathing in about 100 fumigatus spores a day, it's quite crazy to think that 5 of them are probably drug resistant. Don't want to put you off breathing or anything. So I took those 111 isolates to the Public Health England. Sorry, can I just clarify? 2 minutes. Have I got a bit longer than that? Okay, thank you. I was going to say I've got quite a few more slides than that. Okay, so I took those 111 tebuconazole-resistant isolates to what was the Public Health England Mycology Reference Lab in Bristol for CLSI testing to the 4 frontline medical azoles: itraconazole, voriconazole, posaconazole, and isavuconazole. And worryingly, this is the profile of those drug-resistant airborne spores. So I've just highlighted in pink what's classed as resistant according to CLSI. So yeah, 77% of those were itraconazole resistant, 58% voriconazole, 17%, posaconazole, 75%, isavuconazole. So it's, I guess, reassuring that posaconazole is a good salvage therapy for aspergillosis, potentially. But I think what is quite concerning is if you look down at the bottom, this pan-azole resistance in these spores. So you had 45%, half of those spores were resistant to 3 of the 4 azoles, and 14% were resistant to all of them. So what you're actually looking at that in the clinic when someone presents with a pan-azole drug-resistant aspergillosis infection is that they're going to fail treatment with all azole drugs and be put onto an alternative therapy that has higher toxicity and higher chances of treatment failure. So this is what the difference between an air and soil sample looks like. Air is really nice and clean, easy to count. Soil, it's quite hard to actually count how many isolates are on there, so you normally just get a 50 plus. And also that middle plate there, that's the mucorales that Jo mentioned in her talk. That's the black fungus, which is the most common contaminant when you're culturing Aspergillus from environmental samples, and also known as the flesh-eating fungus. It grows at 37 degrees and is always azole resistant. So if you are going to start ever growing fumigatus, always open the plates in the hood, because the mucor's probably nastier than fumigatus. Moving on to the soil samples, got 509 soil samples sent in that grew over 5,000 fumigatus isolates. There was actually 14.2% of those isolates were tebuconazole resistant. And this is just a little plot showing, because I asked people to tell me where they'd collected their soil sample from. And you can see that the majority of azole-resistant isolates are coming from their home compost heaps, followed by the bags of compost that they buy. So just please, if you are going to go and turn your compost heap at home, and then there's those plumes of dust that you disturb from the heap, please wear a mask, and maybe some goggles, because all of those bioaerosols, you do not want to be getting them into your lungs and into your eyes. That's my little public health message for this talk. Right, so Joe mentioned this. I then took those resistant isolates and did a little bit of genetics on them. We look at the CYP51A promoter and coding regions for azole resistance mutations. And what you're looking for is a tandem repeat, often coupled with an amino acid substitution. So I just put this in here because I think it's really cool. When you get those isolates back from Sanger sequencing and you align the DNA sequences for the promoter region, you can see very clearly show up the wild type as a gap, the TR34 at the top there, and then the TR46. And then when you align the DNA sequences and then convert them to amino acid sequences, you can see that L98H substitution that Joe mentioned showing up really nicely there. So for the air isolates, 10% of those 111 actually came back as not being fumigatus. They were Aspergillus lentulus or Aspergillus nidulans, but I kept them in because they can also cause aspergillosis. One isolate came back as just TR34, which is quite unusual. And then the majority came back as TR34 L98H, which expected. However, those 736 soil isolates were incredibly diverse in their mutations. And why is that? That's a question that I would love to find out with future research, because there must be a fitness cost of that plethora of resistance mutations, which means that we see them in the soil, but they don't survive when those spores become airborne. So if you do want to read more about this, there were 3 papers that came out of my PhD. One was basically everyone should use citizen science for their work because they're awesome, and it made my PhD an amazing, amazing experience having those people behind me. One on the soil and one on the air. So very briefly, what has actually come of this work? There's now a Latin American azole resistance survey. I think they've finished collecting spores, but involved 12 countries in Latin America and 26 labs. So I'm hoping that their results are going to become available soon. A group in the Netherlands have taken that sticky sampler and developed it and put those plate seals into what's called a biogrowy. And these biogrowies can hang out for a month, and that means that the air passing through them collecting spores, you can collect spores for a lot longer and ascertain azole resistance over a longer period of time. So we actually have a follow-up piece of work ongoing called MARAF, Monitoring for Azole-Resistant Fumigatus in the UK. And we're asking citizen scientists, farmers, and industrial composters to send us air and soil samples to look for azole resistance, but also to figure out actually where that azole resistance is coming from and whether there are hotspots in the UK. So just briefly, last July, we asked everyone to collect an indoor air sampler in a biogrowy for a month, an outdoor air sample for a month, to bury a cotton strip in their garden for a week, collect a soil sample and a compost sample if they have a home composter. We also asked them to fill out a questionnaire, not going to go into any detail, but just covering things like food bins and how do you dry your washing, how damp's your house, those sort of things that we think influence how many fumigator spores people have in their homes. And this is just preliminary. Really good to see that the results, the plates look very similar to what they did in my PhD. Air sample. Sorry, I think I might have mislabeled that anyway. An air sample at the top and then a compost sample at the bottom. And essentially what we're finding is that compost grows more fumigatus than soil. And also compost grows more azole-resistant fumigatus than soil. And this is what people's home air samples look like, but you might be reassured to see that your indoor air sampler is going to capture much fewer fumigator spores than your outdoor air sampler, and there's going to be, looks like there's less azole-resistant fumigatus indoors than there is outdoors. Right, so that was a little sneak preview. Thank you to everyone who's helped with this research and ongoing work. And that was my happy face after I'd finished processing all of those 8,000 fumigatus isolates. Thanks very much.
Hi, that's good. Caroline Duchesne from Université Laval, Quebec City. Thanks for your talk, very, very interesting. Just a quick question, technical question. You mentioned that you use those sticky covers like for the recovery, but didn't I see that you use like settle plates at the last pictures that you showed that looked like rose bengal agar on a settle plate?
Okay, so actually those sticky plate seals, then to process them, you lay them face down on the agar, and you put them in the incubator and the spores transfer.
So just a question, would it be like, just to simplify, I don't know, would it be possible to just use settling plates like in the homes, or do you worry they would dry out and it wouldn't be usable? And what is the transfer rate when you put your sticky thing on your agar? I'm just curious. It's very interesting. Lots of work, actually. Congratulations.
Yeah, so my concern actually was the agar plates. They would actually potentially start growing fumigatus immediately in the post, and then it would count as potentially a biohazard for postmen. So it completely ruled it out. No, not at all, not at all, because we considered it. And also in the UK, it's a lot more expensive to post a plate than it is a sticky seal, even if you didn't have the biohazard concern.
Hi, very nice talk, very impressive. My name is Ivy Yang. I'm from University of Toronto. I am just wondering if you or your colleagues have tried doing similar analysis using air filter, like indoor filters.
I did do comparative with a Burkhardt air sampler, if you've seen that. We had the impact filter that Joe showed a picture of that was on the roof of St. Mary's, and the Burkhardt that sucks in a known volume of air into tubes of liquid, and then you plate those out. And would you believe it, actually, by placing those air samplers on top of the Burkhardt, you actually got more spores settled on the sticky seals than pulled into the Burkhardt, which was really reassuring for me because I was not just capturing like a percentage of them, but also quite surprising. The only downside, I would say, of using the settle stickies instead is that you can't quantify it as easily because you don't know what volume of air is being pulled through the sampler. And you can go into some crazy analysis of like wind transfer and wind direction, stuff like that. So, yeah. Cool, thank you.